So we've been through interphase and now prophase. So next up we get to metaphase. So during metaphase, your chromosomes are actually going to line up in the middle of the cell. So metaphase is actually kind of easy to remember. You just remember meta and middle. Okay. So here, okay, you can see the chromosomes are really right down the center of the cell. Okay. You have the centrioles on opposite ends. Okay. And then you also have your spindle fibers, which go between the centriole. And the spindle fibers actually connect to either side of the centromere. Okay, so that center point between the sister chromatids. All right, so you get this lining up, and they are all lined up in the same direction. Okay, so you're going to get the correct half of each chromosome getting pulled apart to go into opposite sides of the cell. So yet again, here you can see the cell. The cell is shown by the red. Okay, so the entire cell size is in the red. You've got your chromosomes in the blue here, and then the spindle fibers actually are in green right here. So the sister chromatids will face opposite ends of the cell. Okay, that's specifically how these chromosomes line up. Okay, so remember this side here, the side of the X, is a sister chromatid to this side of the cell here. Okay, so you get one sister chromatid facing each direction of the cell. And then you also have the spindle fibers that connect to both sides of the centromere. Okay, so the spindle fiber from the left here connects to the left side of the centromere. The spindle fiber from the right here connects to the right side of the centromere. So in anaphase, this is where the sister chromatids actually get pulled apart. So they separate to opposite sides of the cell here. Okay, so this is a little bit more difficult to see here. You've got the spindle fibers. You can see they're kind of pulling apart. There is a video that I posted. It's only about a minute long. That is actually showing you in a microscope the whole process of cell division, of mitosis. So you can actually see when anaphase happens, and it happens really quickly. And so from anaphase to cytokinesis, it's pretty quick. The metaphase is kind of like the final stage where it can check to make sure that everything is lined up correctly. So it takes its time a little bit with that. But once it goes into anaphase, it's over. Okay, so in anaphase, you're going to have those sister chromatids pulled apart and then pulled to opposite sides of the cell. Okay, and those spindle fibers are really the driving force behind that, the driving force that helps pull them to opposite sides. Okay, so, and it really helps that the spindle fibers are all kind of moving into the same area, because then you're going to have to reform a nucleus around these chromosomes once you get it over there, which happens in telophase. So during telophase, you're going to get the new nuclear membrane forming around the chromatids on either side of the cell. Okay, so you have this nuclear membrane reforming here. You are getting a distinct nucleus again. Okay, and you can notice that each of these cells now has one copy of each chromosome. Okay. This is also when the chromosomes begin to uncoil again, because they have to go back into their usable format. They're completed with the cell division stage, and so they need to be able to be read again. So they have to uncoil. Okay, so the histones eventually come off, and then you have just this mass of DNA left in the nucleus. You'll also notice that the cell begins to pinch inward. So up to this point, it is still a single cell. Okay, but now you're starting to get that movement inwards that's going to eventually divide it into two separate cells. Okay, and you can see here, you can tell that the centrosome or the centriole is right here in this corner where all of the green lines kind of come to a little bit of a point. And you've also got your blue chromosomes again. Okay, so they are all being kind of pulled into the same area so that it's easy for the nuclear membrane to reform around them. Next up, we have cytokinesis. So during cytokinesis, this is the final stage, and it is when the cytoplasm is officially pinched in half. Okay, so once it's pinched in half, you have two new cells. Okay, the cell membrane completely separates, so they are their own cell, fully formed cell. And that creates the two identical daughter cells. So cytokinesis differs in animal and plant cells, because if you remember, in plant cells, you have the cell wall. Now, the cell wall cannot pinch inward. So plant cells actually form what is known as a cell plate between the cells as they are dividing. So their cytokinesis and telophase look a little bit different. Basically, they have a wall that forms in between the two cells, and the cells are still kind of rectangular in shape. And you get this cell plate there that eventually hardens and produces a new cell wall. Okay, and then the cells will separate, and the cell wall kind of splits halfway down the middle. It also creates the cell membrane on the inside of that cell wall. Okay, so it's a little bit different in animals and plants just because plant cells cannot pinch inward like this. Okay, now if we look at the actual image, you can see that the spindle fibers are no longer very structured. Okay, so they have gone back into really just being the cytoskeleton. Okay, and now you can also see that the green of the cytoskeleton perfectly overlays the red of the cell. Okay, so that's telling you that it has finished up with cell division and it's going back to normal. You also have a very distinct nucleus. Okay, and you can see that there is no condensation of the DNA in the nucleus at this point. It is all back to just an uncondensed mess of DNA. Okay, and you have the final pinch happening here that is separating cytoplasm. Now, while the cell is dividing, it kind of just uses chance to make sure that there are organelles in each of the cells. Okay, because if you're splitting the cytoplasm perfectly in half, okay, chances are you are splitting the organelles perfectly in half as well. Okay, so there's no real big need to go through this type of process that we went through with the DNA in order to separate the organelles. Okay, the DNA is so specific because each cell needs one copy of every single chromosome. And because you have multiple different chromosomes, you have to have a way of making sure that each cell gets a full set of chromosomes. Okay, so I talked about this a little bit. In animal cells, you are going to get the membrane pinching closed until it creates two separate and unique spheres. And then in plant cells, you're going to have the cell plate form between the cells, and then that will eventually form a new cell wall. Okay, so this is kind of looking at an overview of the entire process. So we have interphase, where you just have your normal cell growth, you do not have condensed chromosomes, and everything's just kind of going through the motions like normal. Then you go into prophase. So remember, with prophase, you have the spindle fibers start to form, your nuclear envelope will start to dissolve, and your chromosomes will condense. Okay, because at this point they have already been copied during the S phase of interphase. Okay, which is why you have the sister chromatids connected by the centromere. Next up, we go into metaphase. So at this point, your nuclear membrane has been completely dissolved, your spindle fibers are fully formed, and your chromosomes have lined themselves up across the center of the cell. Then we get into anaphase. So anaphase is when the sister chromatids split. The centromere breaks apart to allow each sister chromatid to move in opposite directions. So here you have the chromatids separating, and you're getting each basically precursor to the new cell has its own copy of every single chromosome. Then you have telophase, where you get the chromosomes start to unravel, so they start to uncondense. Spindle fibers break apart. You have the nuclear membrane is dissolving, and you also have the cell membrane starts to pinch in. And finally, we have cytokinesis. So cytokinesis is when the basically it goes back to normal. Okay, so in cytokinesis you have that final pinching off, so you have two distinct cells, and both of those cells are now in interphase. So it goes from here with cytokinesis right back into interphase over here. Okay, so I know it's kind of weird, the whole process, but you'll get used to it. And then one thing that I kind of find a little bit funny about it is that biology is the only science where multiplication is the same as division. Okay, so you have cell division, but you're multiplying the number of cells that you have. Okay, it's kind of a weird little quote that I found the other day. So anyway, that is the end of chapter 6.2.